Okay, so good morning, everyone. Thank you that you join us for coffee today. Today, our invitation accepted Stjepan Uldrijan. I believe you all know him. He's the research group leader in the Department of Biology, and he also participates in governing our doctoral school, one of our doctoral schools here. Today, we will have a kind of focused discussion about what applied research is and what kind of support you can get here in the medical faculty. But before we delve into that, I would like Stjepan to just say a few words about who you are. What is the main area of research that you are involved in, and what was your kind of journey before sitting in this chair?
Okay, first of all, thanks for inviting me. It's really a pleasure, and I'm pleased that so many of you came today. I hope I won't disappoint you. My name is a Croatian name, yet I come from Brno, so it might be already good to say at the start. So I was born in Brno, and I also did my master's at the Faculty of Sciences, Molecular Biology and Genetics here in Brno. Then I moved to the Masaryk Memorial Cancer Institute, where I did my PhD, or at that time Candidate of Science degree. That was actually awarded by the Charles University in Prague, even though I was there only once for the defense, but fine. And then I moved, actually, during my PhD, I spent nearly 2 years in Germany also. I got a scholarship from the German government, and I participated in the research, which was really nice. Then, after the PhD, I moved to the UK, where I spent 5 years at the Beatson Institute for Cancer Research in Glasgow, which I think is one of the top cancer research institutes in the UK, with really good support and no need to write grants and apply for funding, because the funding is there, which was really nice and pleasant. And then, after 5 years postdoc, I came to Brno, and it was completely different. To apply for grants or try to get some funding, and I've been here ever since. Originally, I was interested in the regulation of the p53 stress response pathways, the famous guardian of the genome, and I studied its negative regulators. So these are proteins MDM2, MDMX that are overexpressed in some cancers to kill the activity of the tumor suppressor. And but in the last, let's say, it's now 6-7 years already, I kind of switched my focus into the biology of malignant melanoma, and specifically into stress response in melanoma, and the cooperation between different signaling pathways to drive the very aggressive growth of the cancer. And that actually is the area where I got the funding from InGA Accelerate to try to find a way how to inhibit the growth of malignant melanoma.
You said 2 important keywords, but I will start with the first one. You said that you study the effect of stress on malignant melanoma. How do you model the stress?
Yes, so of course there are different types of stresses. Actually, the p53 tumor suppressor, that's already a stress response transcription factor that actually stops the cell division and kills the cancer cells or the precancerous cells when they are damaged by UV light, radiation, chemicals in response to DNA damage, but also hypoxia and so on. So that's one type of stress. What we study nowadays is mostly the effect of metabolic stress. So when cells don't have enough nutrients, or the stress when an important pathway that is required for the growth of the cancer cells is inhibited, and they have to cope with that. So we have translational stress project. We also have transcriptional stress project, where by inhibiting transcription, we can actually disrupt a lot of regulatory pathways in the cancer cells, and they really have to work hard to survive that. So.
Okay. You said that you switched your focus during your stay here to study malignant melanoma. What was the driver behind the switch? Was it a kind of opportunity that opened with the collaboration with some clinicians, or was it just the idea that came out, something like that?
Actually, that's quite interesting, because the pathway that we study most at the moment is the ERK-MAP kinase signaling pathway that promotes the strong proliferation of the tumor cells in melanoma. It's driven by mutations of oncogenes BRAF and NRAS in nearly all melanoma patients. So it's very strongly active, and it's very complex with many side ways that can help the cancer cells to overcome the drugs that are used nowadays to treat melanoma patients, to inhibit this pathway. And at the time I was in the UK, there was a group that studied melanoma, or that studied this signaling pathway, not only in melanoma, but also in melanoma. And each time they had a seminar, I was like, okay, come on, this is so complicated. I wouldn't want to do this, you know, this is just a nightmare. I'm happy to work with PT3 and MDM2, which is not so simple, but still, comparatively to that pathway, I thought, no, never. And then at some point we find very interesting response of melanoma cells to metabolic stress that actually impacted on this pathway that drives the growth. And that was really surprising, and that brought us much closer to this signaling pathway, and we started to study it in detail. And when you study something and you go deep enough, you start to understand, right? So once we were confident that we know about the different regulatory pathways, we could come with projects to modify the signaling and see what happens and how it is regulated and what are the feedbacks. So it was really driven by one observation that was very surprising, and that started most of our current projects.
Yes, so this is kind of serendipity that brought you to the field. However, do you get inspiration from the clinical part of research? Do you cooperate with someone who have patients with melanoma? Or do you just work with cell lines, defined cell lines?
Actually, at the time I was at the Masaryk Memorial Cancer Institute, we touched on melanoma, and I actually was regularly going to get the samples of tumors of the metastasis, and they can be quite big. You know, they are like golf balls sometimes, those that are removed. So I had some previous experience, the clinical experience, and I knew this is a nasty disease that is worth spending time. But the manipulations of the signaling that we do, those wouldn't be possible to do in patients, right? We use compounds that are not approved. These are not clinically relevant, really. They are specific inhibitors of some signaling pathways. So for that, it doesn't really make sense to have close ties. Yet we collaborate with teams that study melanoma, different aspects of signaling that are also linked to clinics. One of our collaborating teams is at the University of Dublin, where they have close collaboration with clinicians, and they do proteomic analysis and so on. But we directly don't really use patient samples or do not treat patients. That's something we wouldn't want to do.
Okay, so the major driver of your research is you want to know, you want to understand the process, what's behind the biology, or what's the biology of the process. So this is wanting to know, to understand. So how how can you, this is a kind of preclinical basic research, yes, so how can you bridge the gap between how my findings can actually have some impact in clinical practice and have some, let's say, practical application? Is it anyhow interesting for you?
Mm-hmm. Mm-hmm. So maybe this is how I'm going to get to the current InGA Accelerate project that we have. So melanoma patients are treated with inhibitors of MAP kinase signaling. They work amazingly well. The metastases often go away for some time, and the patient looks like, or the patients look like they're cured. Mm-hmm. Yet in a few more months, the disease comes back, mm-hmm. is now resistant to the treatment. Mm-hmm. And one quite surprising mechanism of the resistance that was described some 10 years ago in a Nature paper was that the cancer cells upregulate translation. Mm-hmm. Specific subtype of translation, CAP-mediated translation, is upregulated by different mechanisms in the tumor cells, and that compensates for the inhibition of this important signaling pathway mm-hmm. that is hit by the compounds, by the drugs that the patients receive. Mm-hmm. So we wanted to see how the translation actually impacts of this signaling pathway, what is the regulation. And we found some really interesting findings, mm-hmm. really interesting data. Surprising, we actually implicated this pathway in the control of the signaling intensity, of the MAP kinase signaling intensity in the melanoma cells with oncogenic mutations. And it was some 3, maybe 4 years back now, where we started to put the data together to write a paper, very interesting paper, hopefully. And while I was putting the results together, I realized that we actually found a new way how to test or how to identify inhibitors of the CAP-mediated translation mm-hmm. of the EIF4F. Now imagine translation, this process. This is not like kinase signaling where you can come and inhibit the module and you see immediately that some phosphorylation disappears. Mm-hmm. In translation, people were looking for inhibitors by radioactively labeling mm-hmm. newly synthesized proteins and testing whether a specific compound inhibits this mm-hmm. process or not. Nowadays, there are some other ways that don't require radioactive labeling. Still, they take a lot of time and they are expensive and slow. And I realized that actually by linking translation to specific signaling pathways, we can look at the response of cancer cells and from that deduct whether the translational machinery is running or not, whether mm-hmm. EIF4F, the translation initiation factor that mediates the CAP-initiated translation, whether it's active or not. Okay, so that was one thing that I realized, okay, maybe we have something that could have practical impact. And second, as I mentioned, this mechanism is responsible for the resistance of melanoma mm-hmm. cells to the clinical treatment. Mm-hmm. So it would be really good to have new inhibitors. Mm-hmm. Yeah? And so I started to think about it, and then we started to kind of develop maybe pilot experiments to see if this system could work as a screening system. Mm-hmm. And for that, I got the funding from InGA Accelerate to develop the screening system. Mm-hmm. Mm-hmm. And it took us 2 years to really have the best combination of reporters and cell lines. And with the support from the faculty, and I have to thank Carolina here and Daniela, who are really amazing. And if you want to apply for InGA, please go and see them. We got really far, and we were confident that we have something that works. Mm-hmm. Yet a problem appeared what to do next with that. Should we patent it and then try to license it to pharma companies who would want to develop new inhibitors? And it turned out that it wouldn't be so straightforward because it would be really difficult to protect this intellectual property because the system can be easily replicated once one has the know-how how it all works. It's really easy to replicate in-house. So, so our project was successful. We got a screening system. We actually tested it in pilot screens in a high-throughput screening system with our collaborators at the Institute of Molecular Genetics in Prague, who are part of the EU Open Screen Consortia for chemical biology. So it all worked, but kind of it went nowhere further because we couldn't do anything more with that. And actually, the InGA, and I didn't mention anything about the funding, so it's not just funding, it's also meetings with mentors. And these mentors are people who have been doing drug discovery research for pharma companies or universities, not in the Czech Republic, but in the US, in Germany, in Switzerland. So people from all over Europe will become your mentors if you have the funding from the InGA Accelerate and you participate in the Spark program. And these people suggested, why don't you actually do the drug discovery yourself? Why don't you find the EIF4F inhibitors to treat the patients, the new compounds? And we said, okay, maybe we should do that. And in the end, we screened, I think, 85,000 small molecule compounds, and we have 200 hits from the screen, chemically very diverse structures. And of those, more than half of them have better activity than the control compound. There is a natural inhibitor of EIF4F that works really well, but doesn't have very good pharmacological properties. But for in vitro test, it's fine as a positive control. So we have a lot of candidates now that we are trying to determine or confirm their activity in so-called orthogonal assays. So other assays, not just the original screening assays, that would confirm the biological activity. And this is actually the basis for the second InGA Accelerate projects that we have running now, trying to move further and prove that some of the compounds could have the activity that we want them to have. And we'll see how it goes.
Yeah, those are amazing achievements. Congratulations.
Thank you.
I would like to go back to the original moment that initiated the journey. You said I started to work with the abrogation of translation as a mechanism of escaping the anti-cancer medicine. And then I realized there is something new, some new mechanism that's not targeted yet, and might be a way how to in the future treat the patient. So, what was the, let's say, what was the decision? I will go the applied research avenue, or if I just publish this and leave it for someone else to go there, what came through your mind?
Yeah, honestly, if there wasn't InGA Accelerate, I would just go on with publishing it and don't even try. I wouldn't even try, to go into the applied research. I mean, that was just my luck that something, this kind of support was available. And again, I stress out, it's not just the financial support for the experiment. This is something that one could theoretically, if one has a really big project, one could kind of as a side way move also the translational part of the research. But for me, the most important bit was the feedback from the people who understand the stuff and who ask the right questions. So that was the driving point, and especially for going further, not just stopping when we have the screening system developed and maybe publish it as a new way how to find these compounds. Because I didn't mention that people in the drug discovery field, they really like kinase inhibitors because they are relatively straightforward to develop and to test. For the translation, it's not so easy, and that's also, and some other mechanisms that were deemed undruggable in the past, such as RAS oncogene, for example, or MYC oncogene. There are much less examples, or many less examples of success in this field. So there are hardly any companies developing compounds targeting these processes. So for EIF4F, there is one company in the US that is openly saying we are developing compound. And this compound is actually based on the natural drug that we used as a positive control. So one limitation for the development is that it's tedious screening, and it's difficult to find new drugs using the traditional way. So I think even if we stopped after the first project when we developed the screening system, it could inspire some further translational research and development in pharma companies using the system that we developed. But as I mentioned, the mentors suggested, okay, you have now an advantage. Nobody knows the system. Nobody knows how to do that. And you can do that quickly and cheaply. And we proved that we can, with screening so many compounds in a relatively short time. And now it's the difficult phase of testing each of them separately in additional assays and confirming that, or picking the best candidates for further development.
Yeah. So applied research is about several different stages of development of let's say, product, and we in the academia are involved in the really early stages to recognize something that might have any potential and to broaden it to a let's say, farther stage where you need to scale up and you need to get really substantial funding to test every individual compound, for example, etc. But even those early stages are meaningful, and if we speak about the support that you get, so this is not only about money to execute your experiments, but you stress out that the importance of the mentors of the Spark program. Yeah. This is something that Carolina brought here to the medical faculty, and this is Stanford University-based network for support of applied research, and this brought in, let's say, experts in the field and from the industry to mentor, to suggest, to select the projects with potential and help to move it further. So this is something that you recognize as a significant part of the support, yeah, the mentors who gave you feedback. Yeah, this is something really important or interesting and worth going farther. Yeah.
Yeah, so I think they really, as I already mentioned, they really ask the important questions. So many scientists, from the experience that I have now after the two Spark mentored projects, many scientists come with amazing ideas of developing new devices or finding new treatments, but they when they start and when they think about going into the translation research and moving towards the commercialization, they don't ask themselves the right questions. Who will buy my product? How much are they willing to spend for that? Will I be able to produce it? And if it's a new device, and I've seen cases where people developed really amazing device, but there is hardly any market for that, even at the universities, in the academia. And it's difficult to have like a business plan that is based on one machine sold in a year. Okay, it goes for really big devices, such as some particle accelerator, but not for relatively small devices. So it's really important, this critical view of the outside experts that tell you, okay, I mean, they were asking the same questions to me. Okay, you have this competing company there; they are much farther than you. They have a collaboration with, I think, Merck or Roche, so they will have all the support. And I said, yeah, but they try to develop one compound that is based on the natural compound, and I now have the opportunity to screen unbiasedly thousands and thousands and thousands of different structures, and that gives me the opportunity to find maybe drugs that will be better, that will have better pharmacological properties, that will be stable, that will be, and we would be able to protect them, the intellectual property. So, for example, in the melanoma treatment, there are I think 3 different BRAF inhibitors given to patients, 3 different MEK inhibitors given to patients. So my opinion was, okay, there is still room for one more EIF4F inhibitor, and as we have everything at hand, let's try it. But still, this critical feedback is important. They ask questions like, how would you test it further? You know how expensive it is. And I said, of course I'm not going to do that. I'm not going to do the in vivo tests. But if we are lucky and we find a compound that is really, really effective, active, has good pharmacological properties in vivo, then a pharma company might be interested in licensing it or buying it, or we could set up a startup to move it further. So there are good examples also at the Masaryk University of drug discovery startups that are successfully moving their findings forward. So, yeah, the critical feedback is important, but it's not to discourage the scientists. It's to support them and guide them in the right direction. Because at least I myself, I get really enthusiastic about many things. Every finding is, I'm really so happy and jumping around. And then, with time, one realizes that it's not that great, or maybe it is interesting as biologically, but the practical impact into therapy, for example, would be 0.
Yeah, how is it to go for a meeting with a mentor to present your ideas? You are not a newbie. You already did something and you got some career, and now you go through this scrutiny and many difficult questions. How does it feel?
No, it's very positive experience, very positive. And also what is encouraging for someone like me who has a project in early stage is the comparison with the other projects, because at the sessions, it's not just one project presented, it's several projects. Every project has, I think, 10 minutes for presentation, and there is some discussion, feedback from the mentors. So one sees several projects during the evening, the presentation, and what was important for me was to realize that we are not far behind the universities in Germany or in other countries. All the projects are at relatively early stage, and that's because they are coming from the academia. They are not coming from the industry and just improving some technicalities on some machine that is already on the market. They are early projects that they have some potential to be successful on the market, but they all require a lot of time. And that's nice to see that we are comparatively doing very well.
I can imagine, that's great. So can you, or is it even possible to generalize what kind of, project are suitable for InGA Accelerate? What kind of goals, data, whatever, who might go this way?
Yeah, so I wouldn't like to generalize too much, because the research that we do at the faculty is very diverse. And so I wouldn't say that drug discovery project is better than a new screening device, for example, for a disease. Not at all, but it's useful really to ask the questions, what comes after the project is finished, after 1 or 2 years of support? What happens when I prove that this is working, that this is a really viable option for treating or testing some disease? What will I do next with that? Will I set up a company? Do I want to do that? And if not, then I think, or if it's not something that can be easily licensed, that I don't see much sense in going in that direction, because it takes a lot of time and energy, and one really has to be prepared that it doesn't stop there with the end of the grant project, but there is a long journey ahead, and also maybe realizing that that there will be these mentors asking those nasty questions about commercialization and who will make the device. Will you outsource it to a company to China? How will you protect the IP? Is it protectable at all? So it's not just about getting funding, but really about thinking about the practical impact of the invention.
And do you get, during the process of Accelerate project execution, do you get the support? Don't publish this, just keep it because it goes the licensing way, or how to protect your intellectual property, etc.
Yeah, I think I didn't mention that, but the CTT also is involved in the InGA projects, and they really have very good insight into what should be done. And I started by saying that I realized that we have something that can be practically, or used translationally to find new compounds. I realized that while putting the data for a manuscript, for a paper, the paper is still not out. We basically stopped doing anything for 2 years towards the publication. We just continued to collect data and try to make the story more convincing and more compelling, but we didn't publish. And okay, it's now in revision, so we'll see if it comes out soon. But at this moment, we already used our advantage to identify the new compounds, and this publication that might someone lead to the screening system is not danger for us anymore. But it's true that it means that the publication will probably have to be postponed if one wants to protect the IP, if one wants to patent anything. Which also means that one should think twice if the person driving that project, for example, is a PhD student who has to finish at some point with publication. In my case, it was in a way easier because the student who was, the primary researcher on that project. She went for maternity leave, so there was kind of interruption in the research. But it's not always that simple, and one has to think twice because students have to publish to finish.
But still, if I think that we recognize also non-publication results for PhD. So this is my follow-up question. What kind of let's say career stage the applied research is worth participate? This was very clumsy sentence. So do you think this is also a way for early career researchers or PhD students to go via the
postdocs or
applied
PhD
research?
students, yeah. I can hardly imagine that a PhD student would come to a PI and say, hey, I have an invention and I will now apply for a grant and run it myself and you just don't bother me. It's usually the opposite way, right? The postdocs and the PhD students have their projects that they have to continue with, finish them. So I think it's important to see that the limitations, for example, the publication, you cannot publish, so how can you finish your PhD if you will be now doing something completely different that is not linked to your project and you will not be able to publish. And even if a patent was accepted as a result, it will take a lot of time, so that's a difference. You want to finish after 4 or 5 years or after 7 years.
You need to finish by
the
4th year.
I think it's nice, and my members of my teams are also here, so they could probably confirm that. I like to give them the opportunity to participate in this project, but it shouldn't be their main project, right? Because that doesn't lead towards the goal that they have, and that's the finishing the PhD studies successfully.
How much capacity does it consume for you?
For myself, not too much, I would say.
Not too much, yeah.
Yeah,
So
because there are people
you didn't interrupt all your other research activities.
No, and as I said, I really enjoyed the meetings of the Spark program. So it's something even positive, I would say. So very, very new experience and very interesting and useful, I would say. So it's something I can only recommend to anyone who thinks they have data results that could lead to some application, clinical application, that they think about it carefully and see what the options are for them, and if they believe it's worth spending the time, and especially if they have an idea what to do after.
What to do after? Yeah.
After finishing that relatively short period, and it's really short. You know, 1 year, 2 years project. It's not enough to develop anything into the stage that you can sell it to a company.
Yeah. And this is an important point. We'll have our ladies to explain the project in a greater detail after our discussion, but still, it's a project, and you need to deliver something to the grant agency. So what do you have to deliver?
So of course, like any other grant projects, it has a plan and it has a timetable of what types of experiments or data will be produced to a certain point, and it has relatively frequent reporting periods. So every 3 months there is a report to be written.
Must be annoying.
I must say, compared to other grants, InGA Accelerate is amazing because the application is relatively short and straightforward form, and so is the reporting. One compares the plan with the with the reality and explains why something didn't work or what to do next, how to go around some obstacle that appeared maybe. It's not too bad at all. Yeah. It's so very friendly, scientist friendly, I would say. So it's great.
It's a kind of, let's say, a tool to track your progress or something like that. But what else do you need to deliver during the project? You need to present.
Yeah, one has to present, and I think that's about it, if I'm not mistaken.
And the outcome of the project?
The outcome, of course, there are different types of results. So the most obvious would be protected IP, so patent, but there are other applied research types of results that come from some classification by the government. For us, it's actually a special class of results called O, from Czech ostatní, the other, which is not the patent yet, but it's going there. We have, for example, in the first project, we have developed the system that has the proven properties that we could use to do the screening. So that was the result, the practical system, the screening platform. That is completely new and much better than anything that was available up till now.
Great. Hey, time is running out, so I would like to touch on a few questions that came from you, and I think that we covered a few of them, but still there are some that I would like you to touch on. And one is about your favorite proverb and how to apply it as a scientist. So I think this could be a good way to start.
Proverbs. It's difficult. Those of you who speak Czech know divadlo Járy Cimrmana, so I'm a big fan of old Czech movies and Jára Cimrman, so I don't really use proverbs. I use these short phrases from the movies, and we use them at home all the time. So I don't even know what is a real proverb, what is not. But if I were to use a real proverb, probably as a recommendation to scientists, I would pick one or two. Maybe it's not all gold that glitters. It's very important to be critical in science. There are many people, not just anywhere in the world, in the best universities, that pretend to be great scientists, and they are not, and their papers are not that great. So I recommend to read from time to time blogs such as For Better Science, or look at PubPeer for potential problems with papers that you have a hard time to reproduce the data. You might spend a lot of time trying to prove or trying to do the same that was published, and then you find that it's actually not possible because it hasn't been done properly. So that's one. And second, and that's really important for young as well as more progress scientists, don't put all your eggs into one basket. So don't rely on one project and on one source of funding, because it is not wise.
And go the way of the applied research via InGA Accelerate.
That's one option. Even though I would say it's not like alternative pathway. It's something that comes as add-on while we are here at the university. In the moment we decide to go purely into applied research, one has to probably choose a different career, set up a spin-off company, I think it's not really like something that could be this or that at the faculty. Here it's more into the basic, basic or less applied research, more clinical research, let's say.
Yeah. These breakfasts are about sharing personal experiences, so there are two more questions I would like to ask. One is, what's the difference between being postdoc and becoming a research group leader? So what's the difference in terms of...
Well, as a postdoc, you have everything at hand. You know, someone gets the funding, comes up with an interesting project, hopefully, that you joined, and it's up to you what you do with these resources and with the time that you have. And so I must say, as a postdoc at the UK, in the UK at the BITS Institute, it was probably the nicest part of my scientific career, because I had no worries, I have no stresses, and I can concentrate fully on the research. And I had the freedom. I think it's important that the postdocs are expected to be independent in a way that they can lead the project in the way they like, you know, with some feedback from the PI, but they are really having the fun of doing science.
Wow.
PIs, it's not always so. Yeah, so the difference is big.
Yeah, but still, is it worth to be a PI?
Yeah, I mean, yeah. If the academic career, you know, and the research is amazing in a way that one can really choose what to do, what one likes, and if one convinces the funding agency that it's worth funding, then it's all great and all plus. Unfortunately, it doesn't always happen that way, and
there's the big if.
Yeah, the grant agencies are not always convinced that this is worth spending their money and tight resources. But so that's the stress of running the group and ensuring the continuity. Yeah. But otherwise, it's a great career.
That's a great career, and it leads to the last question, and this is, how do you handle the stress working in this highly competitive environment and the grant agencies that don't always recognize that you are brilliant and your project is the best?
Well, what can one do? No, I mean, it's not so bad.
It's not so
stress. I think it's also the kind of personal setting. There are people who are stressed always about everything, and for them it might be more difficult. I'm usually quite laid back and not really trying to stress myself about things, especially those that I cannot change. So I don't have any special recommendation. As a young PI, when I started, I used to run here at lunchtime, you know, to relax, always jog in the woods, and that was amazing. Then I got a bit lazy, so I started to go for walks, and now I go for lunch.
So don't skip your lunch. Yeah, this is the final message. Thank you for that. Okay. Stjepan, thank you very much for going to this enterprise, and thank you for participating in Accelerate and all these Spark events, and thank you for sharing your ideas and experience with us.
Thanks for having me, and as I said, if you think you might have something that is worth going into the translational research, that the Spark mentoring and the InGA Accelerate, it's a great way forward, so I can only recommend that. Yeah.
Thank you for that.
Thank you.
Thank you.
So thank you very much for an interesting discussion, but we have few words to say. Since the 3 calls of the internal agency of the Faculty of Medicine are open, so don't hesitate to contact the grant office or to check their website, because startup grants are opened, postdoc scheme is opened, and last but not the least is the scheme LF InGA Accelerate, and our colleague Daniela and Carolina are responsible for it, and Daniela will tell you more about this scheme in which Stjepan successfully applied. So.
Thank you very much. Good morning, everyone. Let me introduce myself briefly. My name is Daniela Podzemná, and I'm a technology transfer assistant and also a contact person for our technology transfer office. And I'm pleased to announce that the next, it means 4th call of LF Accelerate is now officially open. And let's have a look about the key information about this call. LF Accelerate is a project scheme of Internal Grant Agency, and it's inspired by Spark Stanford program, which is a program that of which we are members from 2021. And this program's aim is to bridge the gap between academia and industry. This scheme is designed to assess the application potential of research and development results, and also its goal is to achieve a significant shift in transferring of research results into practical applications. The maximum amount of support that you can apply for is 500,000 crowns, and it's up to 2 years. And the end of the call is scheduled on the 11th of October, and the start of realization of projects, of funded projects, is scheduled on the 1st of January. LF Accelerate has many advantages, but one of them is an ongoing evaluation of funded projects. This includes meetings, quarterly meetings and checklists with representatives from Office for Research and Quality, that means me and my colleagues from Technology Transfer Office. And we also provided meetings with the mentors from Spark that was mentioned by Stjepan. These meetings can help researchers to move forward in their research, and they get valuable feedback from them. These meetings are held twice a year, and twice a year. If you want to know more information about this call, you can scan the QR code, or you can take a leaflet over there on the tables. And on the right side, you can see contact persons for LF Accelerate call, and that's me and my colleague Tomáš Kašpárek. Then I would like to also mention Toucher Sigma, a proof of concept call, which is launched by Technology Transfer Office. So that means it's university-wide call. And the aim is very similar to LF Accelerate project scheme. It's again, it wants to verify application potential of research results, and also it can prepare these results to commercialization. The maximum amount of support here is 3 million crowns for implementation period from 6 to 36 months. And end of the call is scheduled on the 7th of October here, and the start of realization is the same as for LF Accelerate. And I would like to invite you also for a seminar, which is hosted by Technology Transfer Office and will be held tomorrow. And if you want to know more about this call, you can scan again the QR code, and there is also a registration link. A registration link for the seminar. And if you want to consult the the project proposals, you can contact Hanna Pustova from Technology Transfer Office. And that's all for me, and I will pass the microphone.
Thank you very much, Daniela. So, our ladies are happy to answer all your questions according to these two calls after the meeting, and they are happy to help with the application, the grant office as well. And we have another special guest today. So let's welcome Dominik Urban from the Czech Invest Agency, who will tell us something about the support called technology incubation. So,
okay, thank you. Okay. Hello everyone, my name is Dominik Urban, and I am a regional coordinator of technology incubation. And today I would like to give you a presentation about startup support. In particular, I will talk about technology incubation program. I'm from Czech Invest Agency, which is a government body founded by Ministry of Industry and Trade in 1992. And as you can see on the right side of my presentation, there is Czech Invest logo, and I would like to divide it into categorization. First of one is the red one, which is represented by investments, because it was something what Czech economy needed in the 90s. So we focus on foreign investments. Then how Czech economy evolved. We have to change our products and services. So from 2011 we started support startups, and one of the program is technology incubation. Where I see now the biggest role of Czech Invest is where two colors overlap into the dark blue, that we are trying to be a mediator or be somewhere in the middle and try to connect different stakeholders in some regions. We are trying to connect academic sphere with business. We are trying to connect investors with startups. This way. Technology incubation. The main goal of this program is to support 250 innovative startups. We are somewhere half there. We already supported around 120 startups. We still have room for another startups because currently the program is closed, but it will be opened at the end of the year. And the next call also will be in the year 2025. Where we differ from other agencies which are funding startups or funding SMEs, that we are providing systematic support for them. That means that we are offering not only the money and that's it, we are also offering technical mentoring, we are offering networking. We have a lot of data set that we are providing them. Also, we organize events where are interesting speakers. So it's value added for them. What are the key criteria to apply for this funding is especially your project must be innovative. It means that it must be unique, at least on a national level. It must be feasible. That means that your activities must be described logically and must to lead to the target goal of your project. And last one is that your project must be scalable, and that is simply that your product, your technical product, is easy to adapt on different markets. Or change in demands on that market. Minimum level, technological readiness level is 2, so it means that we cannot support something what is only in your head. it must be written down. So you need to provide us with some technological documentation or with some process of thoughts and so on. We support startups or spin-offs, which are 3 or 5 years old, maximum 3 or 5 years old. And you have to be based in the Czech Republic. At the end of my presentation, I would like to showcase some already supported projects. One of them is e-cafe bike. These all projects are from South Moravian region. E-cafe bikes are developing premium electronics cargo bikes. Another interesting project is Sensoria. These guys are from Brno, and they are developing smart greenhouses, which operate the whole year, so they can always provide you with fresh vegetables. Another interesting project I picked is Miko, and they are developing packaging which is created from mushroom materials. And last one, I picked Kikiriki Games. They are gaming developer, and they specialize on mobile devices, and they produced a mobile game for blind people. And my last slide is about data. On the left side, you can see a map of Czech Republic, where are different regions, and these numbers means numbers of supported projects. So you can see in Prague, the first one is Prague with 61 projects. Then there is a trend, big step down. South Moravian region, we are second with 35, and the other regions are with only few projects. And on the right side of my presentation is industries that we support. So you can see that AI is the most supported projects are from AI industry. Second one is creative, which is video gaming, and Tech for Life is, for example, somewhere at the end, which Tech for Life means healthcare industry. And that's it. So if you have any question, you can ask me. Thank you.
So thank you again, once again, for attending us, for joining us today. And let's chat, let's grab a coffee, let's grab a croissant, and we are happy to answer your questions now or later, but not until the September of 11th, at the latest. Thank you. Have a nice day.